This integrated research platform focuses on isotope and chemical analysis. We use subtle changes in isotopic ratio to investigate phenomena related to biological and environmental research. We use both isotopic tracers and natural abundance measurements to quantify rates of chemical transformation, identify where compounds and reactions are taking place, and identify the organisms linked to these processes. These data complement other types of analysis to provide insights for tracing soil carbon, understanding microbial interactions, informing atmospheric and climate models, and many other types of applications. EMSL has successfully used isotope and chemical analysis to develop state-of-the-art techniques that can collect a wide range of interesting data. For example, we developed in situ liquid SIMS, or liquid secondary ion mass spectrometry. This technique blasts a high-energy ion beam at a sample surface to ionize molecules suspended in solution. The ions can then be separated from the solution and detected by a nearby mass spectrometer. For instance, our team recently used liquid SIMS to describe the complex initial nucleation processes of secondary organic aerosols. Another of our specialties is chemical and isotopic imaging. The high degree of spatial resolution of our approach, on the order of nanometers, is possible through a technique called nanosims. This allows us to trace nutrients at intra- and intercellular scales, as well as between organisms and their environment. For example, we can trace nitrogen fixation from bacteria into plants to study the productivity of plants in nutrient-deprived soils. As we look to the future, this integrated research platform will study biogeochemical processes that are not just occurring at large spatial scales, but are truly driven by localized microbial, geochemical, and biotic processes, including things like plant-soil interactions and cell signaling amongst microbial communities. We are working to bridge these quantitative approaches from the molecular to landscape scales to better enable a more holistic accounting of biogeochemical transformations within a system. We provide measurements that can be integrated with data from other platforms to help better understand complex systems at multiple spatial and temporal scales. Our goal is to develop predictive models which will help us better understand our planet from the molecular to the global scale. This is critical to ensuring a sustainable future.